All right. Thank you very much for joining us today. We have Stephanie Seneff, who is a senior research scientist at MIT, and she is very much involved in public health and the chronic disease epidemic. I'm very happy to have Stephanie here today, and she's a big inspiration for my work. I just want to give the audience some background for this conversation. And she has done amazing research on the connection between deuterium and cancer. And she inspired me to look into cancer more deeply and question my own views on cancer. And without her work, I would have never had the inspiration to view cancer in a different way, as basically a defense mechanism against toxicity. And so I just want to highlight that Stephanie's work had a tremendous impact on my thinking. And if it was up to me, she would get a Nobel Prize for her cancer work. But for this interview, it's meant to basically bring together two scientific minds. Both of us are scientists, and kind of explore the scientific discovery process. We're going to get into how is it that Stephanie came to this new view of cancer, and how it led her to think about cancer in a different way. But before we get into that, I just thought we would touch on your background, and because oddly enough, we do have similar backgrounds, both coming from the physical sciences. And so, do you mind, Stephanie, just talking a little bit about your scientific background, and including your background in computer science and how you got interested in the chronic disease epidemic?
Sure, thank you. It's great to have me here today. So my history is kind of interesting. I started out in biology. Biology has always been my big passion. Undergraduate degree from MIT in biology way back when, and then I went on to graduate school, but I switched careers at that point and went into computer science, which was a big change, and I got my PhD in computer science along with a couple other degrees, master's and EE degrees. From MIT. I've been at MIT, by the way, all my adult life, ever since I was 16 years old. So I know it well, and I'm still there. So it's kind of a miracle. Anyway, so I got this degree in computer science. I got a job at MIT, and my work was involving training computers to be able to communicate with humans using natural language. That was the core of my work, and fascinating stuff. I wrote a lot of computer code. We were pioneers in the space of we probably had the very first human computer dialogue system possibly in the world way back when. You had to go have a cup of coffee while you waited for the answer because it was so slow. But we knew that things were going to speed up in the future. I always kind of thought we were wasting our time because I just never thought that technology would get good enough to be useful, but I was wrong. And so that field really took off. So it was about 2007, after many years of writing code, training computers, that I got interested in autism. I was seeing the rates go up every year. We're just diagnosing it more. And I thought, no, that's not right. Something's going on. And I wanted to figure out what it was. I thought, well, I can do some analysis of correlations between the rise in various chemicals that the kids might be exposed to, looking for a correlation with a rise in autism. And I found out actually many other diseases were also going up dramatically over time in that first decade of this century. So I started analyzing the data online and came up with incredible correlations of rises in various diseases, not just autism, and the rise in glyphosate, the active ingredient in Roundup. And I didn't really think of glyphosate at first. I actually studied for 5 years autism, looking at things and ruling them out and kind of coming up short. I thought maybe the vaccines had something to do with it, and that was about it, And then I thought maybe it's something they're eating in the food because they have digestive problems. So I was kind of looking for some chemical that might be in the food. I wasn't thinking of glyphosate. It's safe. People use it all over the lawns or for the dandelions. It's supposed to be extremely safe for humans and the best herbicide on the planet, the most used herbicide on the planet. I didn't think of glyphosate, but I went to a concert, really an epiphany moment for me, 2012. Professor Don Huber gave a 2-hour presentation on glyphosate. When I walked into the room, I didn't know what glyphosate was. Of course, I knew about Roundup, and I never used Roundup because I don't like to use toxic chemicals. I was really spellbound in that lecture because he was showing all these different ways in which glyphosate affects the soil health, the microbiome of the soil, the microbiome of the gut, the liver enzymes. I mean, there were all these things that glyphosate causes. He was an expert. He is an expert. He's still active on glyphosate. And he really opened my eyes. I finished that. I walked away from that presentation convinced that I had found my answer. So I went back and studied everything I could about glyphosate. And the more I studied, the more convinced I was. I mean, it was really amazing. And then we found, Nancy Swanson and I collaborated on a number of papers. We found incredibly strong correlations, between autism and glyphosate. The rise in glyphosate usage corresponds exactly with the rise in autism in the US. And then there's a whole bunch of other things, conditions, diabetes, obesity, various adult neurological problems like Alzheimer's and Parkinson's disease, of the aged. So the elderly and the children are both affected, I think, by glyphosate to cause neurodegenerative disease. And then there's also various cancers like pancreatic cancer, liver cancer, kidney cancer, thyroid cancer. They're all correlated, so it's an amazing list. And I became convinced that glyphosate is a very special chemical with a very unique mechanism of toxicity that I identified as its ability to substitute for glycine during protein synthesis. I believe that's happening. The industry denies it, but I believe that's happening.
Right. And we'll get to deuterium and cancer later, but since you brought up glyphosate, why don't we focus on that at the moment? And just if you can, very succinctly, try to explain to the audience, what do you think is the molecular mechanism by which glyphosate manifests its toxicity?
Yeah, and I think a key issue is the gut microbiome. Glyphosate has been patented as an antimicrobial agent. It's been shown experimentally to suppress critical microbes in the infant gut, lactobacillus and bifidobacteria. They're really central to the health of the baby's gut. Glyphosate suppresses both of them. They're very sensitive. And then other microbes like Clostridia tend to overgrow, and you get into a situation with too many pathogens in the gut that causes inflammatory bowel disease, which is one of the diseases that's going up dramatically exactly in step. And then that ends up messing things up even more because you've got that inflammation, which causes certain critical microbes that required an anaerobic environment that's not there anymore because of the inflammation. So they die off and you get into a downward spiral. And, all these different gut problems that we have, including celiac disease, by the way, which I think is a direct hit. Gluten intolerance is an epidemic now. So many people are eliminating gluten from their diet. Gluten has a lot of proline, and proline is an amino acid that's difficult to break apart in the chain. You need specialized enzymes, and lactobacillus produce those enzymes for the host. So when the lactobacillus are down, those enzymes are not adequately expressed, the gluten doesn't get digested, and then you have these short peptides that are allergenic. And you end up with, and then you develop antibodies to those short foreign peptides that circulate in the blood because glyphosate sets up a leaky gut barrier. And then those antibodies actually, they attack human proteins in the thyroid that have a similar sequence. And that gives you the thyroid disease, which is another condition that is going up with glyphosate usage.
Yeah, so I've been interested in the possibility that some of these autoimmune diseases might be misinterpreted, that maybe they are toxicity-based diseases. I don't know, is there any thoughts that you might have on, because you just mentioned autoimmune?
Well, I do, yes.
Yeah.
In fact, I do think they are. And of course, we have to bring up deuterium now because that's central to my most recent studies. And let me tell you, I had another epiphany, 2019, December. I remember very clearly, Laszlo Boros sent me an email message. I didn't know who he was, a blind email. And he said, he congratulated me on a paper I had just published. And then he said, by the way, do you know about deuterium? And I knew what deuterium was, but I had no clue that it had anything to do with biology. We had a web Zoom call interaction, which was really fabulous. And I just got on fire. I was another, I really give a lot of credit to Laszlo Boros and to Don Huber for impacting my future directions in really changing the course of my research. So that was December 2019. I jumped right on it. Again, I started reading everything I could about deuterium. I immediately saw the connection to glyphosate because glyphosate just, suppresses the microbes in the gut that supply deuterium depleted nutrients to the host. So that's a direct hit. Basically, these microbes digest fiber, for example, and they make hydrogen gas from it. And then they use the hydrogen gas to make short chain fatty acids. And those short chain fatty acids are extremely important nutrients made only by the gut microbes. The human cells don't make those. And then the short chain fatty acids, especially butyrate, are fuel for the melanocytes. And the butyrate keeps them very happy. It's like their number one choice for fuel, like 70%. Ideally, 70% of their nutrients are butyrate. Butyrate is low in deuterium, I believe, because it comes from that hydrogen gas. And the process of making that hydrogen strips away 80% of the deuterium. So deuterium is heavy hydrogen, just to get, some people are probably lost at this point because they have no idea what it is. It's heavy hydrogen. It's rare compared to regular hydrogen. It's like carbon-14, right? It's a different a different form of the atom that has an extra neutron. So hydrogen is the smallest atom. It's on the upper corner of the periodic chart. 1 proton, 1 electron. Deuterium is 1 proton, 1 electron, and 1 neutron, which makes it twice as heavy. It's present in seawater at 155 parts per million. So that sounds small, but it's present in our blood at a level that's 5 or 6 times as much as calcium. So compared to the minerals, it's very high, even though compared to hydrogen, it's very small, because hydrogen is by far our most common atom in our body. I mean, most of our atoms are hydrogen atoms, you know?
Yeah. Speaking of which, I do occasionally drink deuterium depleted water. I think I saw an interview where you also do that as well. Just a random question. Have you noticed any benefits from drinking that yet? I know it can take some time.
Yeah, I think so. I
Okay.
mean, I've been drinking it now for 2 or 3 years, every day, a small amount, just 1/3 of a cup, which I mix with 2/3 cup of water. And I've just been doing that every day for a long time. It's expensive. It's crazy expensive compared to water. But if you compare it to wine, it's not so bad, you know. So, yeah, and I don't know, but I'm 78 years old. I don't take any pharmaceutical drugs. I don't have any aches and pains. I mean, I'm doing pretty well. My brain's pretty sharp. I would say I'm doing well, you know. So
Yeah.
I've never really had many health problems. I've always been against chemicals, you know. Like I won't even spray a roach, you know, with a chemical. So I think that's helped to keep me healthy.
Absolutely. Yeah. And that brings up a deeper issue, which is that every time, and I think this sort of concurs with what you're saying, but every time I've looked into different diseases, you know, Alzheimer's, autism, Parkinson's, et cetera, and I went and read the medical literature on it, there was always a connection to toxicity.
I know,
Every single
absolutely.
one.
I totally think toxicity is a reason why we're so unhealthy right now. And our country is very loose with the rules, you know. We have very poor regulatory constraints compared to Europe. Europe, bans or restricts many pesticides, many toxic chemicals that we're fine with them, you know, like no problem.
Yeah. And, you know, and they even, it's shocking to think that we're in a first world country, and yet, you know, those of us who care about our health have to filter the heck out of our water. Like,
I know.
how is it that we don't even have clean water?
Yeah, and it's hard to find a place to live where you're not exposed to somebody else's toxins that are being pushed into your face and you can't stop. You know, it's very frustrating. Even if you have an organic farm, probably have a bunch of farms around you that are spraying glyphosate, you know, and other chemicals.
Yeah.
So it's really frustrating.
Yeah. You know, since you've mentioned both glyphosate and deuterium, you know, and in my own work, I've also looked at some other toxins. And of course, I know I follow you on social media, so I know you're outspoken against things like heavy metals and vaccines and things like that. Do you have a sense for what you think are the most important toxins, if you had to kind of like list a couple? I mean, obviously, I would imagine glyphosate's up there.
Glyphosate's right there at the top. Yes.
Okay.
Aluminum, glyphosate, you know, lead, arsenic, all those toxic metals. I mean, the heavy metals.
Yes.
Those are all a problem. And of course, all the pesticides, you know, the insecticides, herbicides, fungicides, they're all bad. And we use them heavily. And in fact, we have to use more of the insecticides and the fungicides because of the glyphosate. Glyphosate makes those plants sick, so they're not as resistant to those other assaults. And so we're using way too many toxic chemicals on our crops. And you know, the government has this attitude that, oh my God, we can't live without glyphosate, we'll all starve, you know, so they're just not willing to regulate it. And that's a mistake. I mean, we really need to. I really applaud any young person. Right now, I feel if you're a young person starting out today, buy a small organic farm and make it work, you know? That should be #1 priority for anybody who's trained to write code, and now code is useless. Go buy a farm. You know what I mean? Because the coding is being taken over by the computers. Those kinds of skills don't count anymore. So pivot. I think it would be wise for young people to heed that call and really start to open up these small organic family farms. We need to repopulate our country with those, I think.
I totally agree. Yeah. And we also need more ranchers raising animals. And,
absolutely.
Yeah.
Yes.
So you did touch, I think, a little bit, or hint at politics a little bit. So why don't we just mention that glyphosate was in the news this past year. It was in the news for negative reasons, in the sense that I think public awareness was growing around the negative impacts of glyphosate, and yet at the same time, the government made a certain bad decision, I think, that we're well aware of. Do you want to just speak a little bit about the government decision that was made around glyphosate?
That's terrible. Yeah, it was a Missouri case where they won a trial of non-Hodgkin's lymphoma. There's like 100,000 people waiting in the wings who think that glyphosate caused their non-Hodgkin's lymphoma. And the first one was Dwayne Lee Johnson. He was in California. He was a school groundskeeper. His job was to control, keep the school grounds nice. And he used exclusively glyphosate, which is already a problem. Glyphosate on the schoolyard is really bad for the kids, right? And he got he had a couple of young boys. He got non-Hodgkin's lymphoma in his early 40s. And so he put through this lawsuit, and that was the first one. I remember following it and thinking it's never going to work. They always have all these, really hot shit lawyers, and they're going to just shut it down. But he succeeded, and he got a very big award by a jury trial. It later got reduced to a much smaller amount, which is what typically happens. But this Missouri one was another case. There have been several now since then that have succeeded. And as I said, 100,000 people waiting in the wings. So this was a big deal. And to me, and this was why Bayer bought Monsanto. Monsanto was the original producer. Back in 1974 was when they first put glyphosate on the market. It really picked up starting at the turn of the century when they put out all these GMO Roundup Ready crops. That's the really big deal. Core crops of the processed food industry are all sprayed with glyphosate routinely. The crop doesn't die. The weeds die. It's a great system for cheap farming, because you don't have to hire a bunch of people to pull weeds. That's the big trick with it. And anyway, so he won that lawsuit, and then some other people won lawsuits, and then there was the Missouri case, and the Bayer, who now owns Monsanto, they took it to higher levels, and they took it all the way to the Supreme Court. And what happened this past week is the Supreme Court killed it. They argued in favor of the industry, and said these people don't have a case, no deal. And that is devastating, because that means all these 100,000 people that are waiting are dead in the water. They're not going to be able to. They'll have to have an incredibly strong case to win, and lawyers will probably say, I won't even help you. It's just hopeless, So it's a big, big win. They'll probably put Roundup they'll put glyphosate back into the Roundup now, probably, because they decided not to sell glyphosate in Roundup to the American public, the people who use it for the dandelions. They were happy to put the Roundup the glyphosate in the version of Roundup that's used on the crops. So no problem with the crops, because the farmers can't prove it. They've always got so many other things going on that they can't prove it. So even if they get non-Hodgkin's lymphoma, they'll say, well, it was that other chemical you were using. It wasn't the glyphosate. So it's only the public that won by virtue of them taking the glyphosate out of the Roundup. Of course, they put a bunch of other toxic chemicals in there, which probably are considered to be worse than glyphosate. So it's not really a win even there. Right. So we need to go back to organic farming. I think it's essential. Regenerative organic farming. Fix the soil. Right now, the soil's wrecked because the glyphosate's been killing off all the soil bacteria, taking out all the minerals. I mean, it's really in bad shape. And the plants are much more sensitive to drought when there's not enough rain. I mean, the whole thing just falls apart because of these toxic chemicals. So we have to think in terms of nurturing the soil and nurturing the crop rather than poisoning it as a proper way to grow food. And that will keep us healthy as well, because the food we eat will be healthy. It's absolutely essential that we need to do that. And of course, the government's working the other direction. And Bayer has been very hard at work trying to get various states, individual states, to deny, to pass laws that would protect glyphosate from any lawsuits. And so, and they've been working at the federal level for that as well. So they're working really hard. They're spending lots of money on politics to assure that glyphosate stays on the market.
Yeah, that's really tragic. And, for people in the audience, they need to know that those of us who've really dove deep on chronic disease and the origin of chronic disease, we have found that toxicity is at the center.
Absolutely.
And so that's what the message that I would share with the audience is that this is a big deal. And, we need the government to listen to the people that have dove deep on this topic, listen to the true experts, the people that have really study this,
Yes,
and we need the government to listen to us. It's really a shame.
we really
There's
do.
obviously a disconnect between us and the government.
Right.
Yeah, but yeah, thanks for sharing that. I thought we could also maybe move over to deuterium now.
That would be great.
Yeah, and so when it comes to deuterium and cancer, which one did you start looking into first? Would you start first with deuterium and then get into cancer, or was it vice versa?
I was interested in cancer from way back. I've been interested
Okay.
in all of these diseases: Alzheimer's, cancer, heart disease, autism. I've been interested in Parkinson's disease, all of them. I've been fascinated by them. And when I read the literature, I understand that they don't understand what's going on. I mean, it's pretty clear, right?
Mhm.
There's a lot of mystery around all of those diseases.
Mhm.
And that's been frustrating to me. And I have to say that, when I was looking at, I love to look at pathways and enzymes and, where they're located in the cell. I'm so fascinated with metabolism and how it works and why. I'm always asking why, And so I saw this situation where there's these mitochondria, and this is all before I knew about deuterium. The cells have lots and lots of these mitochondria, these little organelles inside the cell. Mitochondria make the ATP, which is the energy source for the cell, using an enzyme called ATP synthase. It's a nanomotor. It actually spins around like 500,000, 50,000 times. It's a ridiculous amount of spinning that it does in a hurry. And it does that from a proton motive force. There are protons that are coming through the motor and spinning it, like a water wheel. And those protons are very important. And the mitochondria are set up to pump protons into the intermembrane space. And then those protons are forced to come out through those motors and spin them. It's a beautiful system. But what I had seen was I was curious about where are enzymes located. This was long before I knew about deuterium. I was just puzzled because I was looking at cholesterol, and how the liver cell, it gets the cholesterol. It transports the cholesterol into the mitochondria, and then it has enzymes in the mitochondria that oxidize or hydrolyze the cholesterol, change it into hydroxycholesterol. Then there's and they have to transport the enzyme into the mitochondria and the substrate into the mitochondria. You do the reaction there, then you transport the product out of the mitochondria, and so it's like, well, why does it go through all this extra effort to do the reaction in the mitochondria? Why not just do it in the cytoplasm? I was really puzzled by that. It was a question I had. And I thought, well, my metabolism always has a reason. Everything biology does has a reason. So there must be a reason. And then as soon as I learned about deuterium, I said, of course, that's it, And it turns out there's lots of substrates that are pulled into the mitochondria. And then they have these enzymes in the mitochondria that produce deuterium depleted water from those substrates. And it's really fascinating. Cholesterol is a deuterium depleted nutrient. And I feel that's true because I can see how it comes from acetate. It's like 3 acetate molecules are the basis. And there's a whole bunch of steps. It's a very complicated reaction. Finally makes cholesterol out of these 3 acetates. But the acetates, if they're produced by the microbes, are going to be low in deuterium because they come from that hydrogen gas. And the hydrogen gas is 80% down. So it's really, really critical, that hydrogen gas that's produced by the microbes. And the microbes that produce that hydrogen gas don't like reactive oxygen. And the inflammation that's generated by glyphosate and other chemicals causes reactive oxygen and sets up a situation where they can't make the hydrogen gas anymore.
Okay.
It's that simple.
This is super fascinating. I wanted to just dive into that point that you mentioned about cholesterol. So are you saying that the cholesterol that we make inside of our own bodies is deuterium depleted?
I think so. And I don't have a paper that says that. I have to confess, I don't have a paper, but I just based on I'm following the pathways, because it starts out with 3 acetate molecules. And acetate is it's the shortest short chain fatty acid. It's produced by the microbes from hydrogen gas. It actually reduces carbon dioxide using hydrogen gas to make acetate. There are acetogenic bacteria that do that. So that acetate's going to be 80% down because its hydrogens all come from the hydrogen gas. It's very logical. And that's why those short-chain fatty acids are so special. Acetate gets converted to butyrate through a couple steps in the microbes as well. They make butyrate from the acetate. So the butyrate's also depleted in deuterium. And that's what the colonocytes consume as their fuel, as their primary fuel.
Okay, this brings up another interesting point, which I know is related to your work, but things that are made by the body can sometimes be naturally deuterium depleted. But if we try to synthetically take a supplement,
you are right,
that would replace that, it might not be deuterium depleted. Can you elaborate on that and how it shows up? In practice?
Absolutely. In fact, I have a paper that I published that talks about that, because it's quite fascinating. I was really amused by studies that were done where they used choline. So choline is a very healthy nutrient found mostly in animals, eggs, for example, seafood. Lots of choline, grass-fed beef. It clearly is a beneficial nutrient. From evidence that people have seen. However, so choline has a nitrogen atom that has 3 methyl groups attached to it. Trimethylamine is a part of choline. And the gut microbes can convert choline to trimethylamine as a single unit. So it's CH3,3N, if you know what I mean, 3 methyls attached to a nitrogen atom. And trimethylamine is a super interesting molecule. And I've written more than one paper focused on trimethylamine, because it's solely produced by the gut microbes. If you don't have, like they have these these mice that are that don't have any gut microbiome, these germ-free mice or something, what they're called, they don't make trimethylamine. So it's clearly the microbes that are doing that. And then the trimethylamine goes on to get further metabolized by these anaerobic bacteria. And it gets metabolized. The methyls get converted to methane gas, and that's all going to be deuterium depleted, assuming the choline is. And the choline would be deuterium depleted if it came through a natural process. But the interesting thing is that people take choline as a supplement because it's supposed to be so good for you. And what they take often is choline bitartrate. Choline bitartrate. It's a synthetic choline. So it doesn't have the depletion in deuterium. And what was really, really fascinating is they did this study on mice where they fed them fully deuterated choline bitartrate. The methyls, they have CH3, that's 9 protons in there with 3 methyls. All of them were deuterium. They gave them this choline that was marked with deuterium. They did that so they could trace the deuterium. And but they also inadvertently did an experiment to say what happens if the choline has enormous amounts of deuterium in it. What happens then? And they found really bad stuff. Like there's this molecule called trimethylamine oxide that's produced from trimethylamine. It gets shipped from the gut to the liver. The liver oxidizes it, and it makes trimethylamine oxide. And if you look at the literature on TMAO, you will find an astonishing number of papers that show it's just like glyphosate. It's like connected. It's related to all these different diseases. High levels of TMAO in the blood are bad news for a huge list of diseases. And so I was fascinated by that. Actually, I learned about TMAO a long time ago, and I'm like, I don't get it. This is weird. I don't understand this at all. And I walked away. I couldn't explain it, Now I can. So it's so cool that the deuterium has caused me to fill in gaps in my theories. And suddenly everything makes sense once I realized the deuterium situation. So when you feed so they found that the choline bitartrate, and they found this for humans too. They fed the humans deuterated choline, which is a horrible, horrible thing to do. They had no idea that deuterium was toxic. They thought it's just great, we can mark it. When they fed it, I think it was the human study where they saw that it showed up, the trimethylamine oxide showed up in the urine. None of the choline got into the feces. In other words, the microbes got rid of everything that had deuterium and shipped it off to the liver. So what I think is happening is that the TMA that's in the gut, the microbes actually metabolize it using enzymes that know how to ignore it if it has a deuterium on it. So they actually have these fancy enzymes, which are so cool. Not just the microbes, we have enzymes like that too, that they won't react. If they have a substrate and they're about to turn it into something else, and they want to pull a hydrogen off of it, and it's a deuterium, they say, oh, no, go away, I don't want you. They won't do it. So there's like a deuterium kinetic isotope effect. It can be really high for the conversion of, for the reaction to take place. So the enzyme has a special, and it does this through proton tunneling. It's really clever. The enzyme sets up a situation where the proton has to go through a skinny space, and the deuteron can't fit. So it keeps it from reacting if it has a deuterium atom attached to it. So the microbes wouldn't touch the TMA if they came from the deuterated choline. they wouldn't touch it. Whereas if it didn't have any deuterium in it, they could convert it into really useful things, because it can convert it to methane and to formaldehyde, both of which are going to be low in deuterium if the source was. And also because there's that filter that they don't like to do it if there's a deuterium already. So they're filtering out the bad guys, shipping them off to the liver. So the TMAO becomes enriched in deuterium because what's left behind, is enriched. So TMAO becomes a marker for both gut dysbiosis and deuterium toxicity in the methylation pathways.
Right.
Methylation pathways. So they said that they had eggs, they had humans who ate tons of eggs, and they had humans who took this deuterated TMAO, deuterated choline. The ones who took the deuterated choline, the TMAO levels went through the roof. The ones who ate the eggs, no problem at all. Nothing.
Fascinating. Okay, so if I can just try to add my 2 cents or summarize here. So basically there's this molecule that people have connected to certain chronic diseases, TMAO, and or trimethylamine, right? Or whatever it is.
TMAO, the one that's
TMAO, TMAO. And you're interpreting that as a marker for deuterium overload or deuterium excess. And so it really can be correlation, not causation necessarily, but whatever. Basically, the idea is
the molecule is acting as a signal to say, hey guys, you got some trouble here. You got to get rid of the, and what it is excess deuterium in the methylation pathways. And the methylation pathways are really cool. And so people know about methylation, the methylation pathways and how important they are for health and blah, blah, blah. What they don't realize is those methyl groups are severely depleted in deuterium.
Right.
If they're natural, but this choline bitartrate that people take doesn't have that filter. So you're dumping excess deuterium into the methylation pathways.
Yeah, and so just for the audience, if you're listening and you're deciding between eating steak and eggs for breakfast or taking a whole bunch of supplements that would be equivalent to the nutrients in the steak and eggs, you should eat the steak and eggs.
That's right. That's right. And I tell you glycine as well. So glycine actually, it's the smallest amino acid. It has just the one carbon in the core. And that carbon is deuterium depleted because it comes from formaldehyde. Glycine is a carrier of a methylene unit that can be fed to folate, methyl tetrahydrofolate. You probably don't know the chemistry, but methyl tetrahydrofolate is made by the microbes, and the methyl is passed over to homocysteine to make methionine. And so methionine is a universal methyl donor, and that's an amino acid. So that methyl that's attached to the sulfur atom of methionine came from methyl tetrahydrofolate, which can come from glycine. And so it actually makes glycine from methyl tetrahydrofolate, and it converts glycine to methyl tetrahydrofolate. Does that make sense to you? Glycine has that methyl unit as its core.
I see.
So that's pretty cool. So glycine is actually a low deuterium nutrient if it's natural. But of course, all the glycine you take as a supplement is not natural.
Very interesting. Okay, I have to check my magnesium glycinate. And maybe stop taking that. But no, this is a great point. And I have been trying to gradually wean off my supplements as much as possible and just become more and more natural. And just so the audience knows, Stephanie's research is providing the theoretical justification for doing that, that supplements are not equivalent to real food. So I thought we would just now switch over to deuterium and cancer. And so I just want to add my own personal note that I first came across your theory of cancer when listening to an interview with Anthony Chaffee. You did an interview with him. And in that interview, you presented the connection between deuterium and cancer. And when I heard that, I was mind blown. It was totally shocking for me. And I think it was also very different from what Anthony Chaffee had heard in the past. So it was probably a little bit shocking to him as well. But yeah, maybe just walk us through this new, different interpretation of what cancer cells do with deuterium.
Yeah, it's incredible. And I really feel like we're onto something absolutely huge. And I've actually published several papers now linking cancer to deuterium. So I'm very happy about that. More than, I think it's maybe 3 or 4. I think I'm actually losing count. But I'm very excited because I think we've really hit pure gold here, because it's clear to me that the cancer cells, they do 2 things. They primarily do 2 things. They hoard deuterium. They keep it internally, and they reproduce. They grow bigger. The tumor grows bigger. They're really obsessed on two things, right? Hold on to the deuterium and produce deuterium depleted nutrients to feed to the world. So they export a lot of things that are really good sources of deuterium depleted protons, including actually protons that are deuterium depleted. So what's really interesting is that the cells this is maybe the strongest example of the evidence that cancer cells hoard deuterium, because the cells, all the cells, they have these lysosomes where they digest, they break down broken molecules and recycle them into new materials in the lysosomes. It's their digestive system. And they have these ATPase again, but these are opposite from the ATPase nanomotors in the mitochondria. They actually pump protons. They use up ATP and pump protons into the lysosome. And those protons are not deuterons because those enzymes have a very high deuterium kinetic isotope effect. So they're pumping the lysosome full of high quality protons. And the lysosome is going to break down big molecules like proteins into individual amino acids and various complex sugar chains into individual small molecules. Every time they break apart a bond, they got to put two protons there. So they want those protons to be deuterium depleted so that those products will be deuterium depleted. So it's important to make sure the lysosome has low deuterium. And they do that through these ATPase pumps that push protons in. Well, what the cancer cells do, they take those pumps and move them to the membrane. They move them to the plasma membrane. And then they pump the protons out into the world, into the external space. So they are actually actively pumping out of the cell deuterium depleted protons, leaving behind deuterium enriched protons, right, inside the cell. So they're increasing their deuterium content in their cytoplasm. And meanwhile, they shut down their mitochondria. Even when they're healthy, they say, okay, we're not going to make ATP using mitochondria. We're going to let them rest. And instead, they make the ATP using glycolysis. So they have, in the cytoplasm, they take in tons of glucose and they process it through glycolysis. They get a tiny bit of ATP out of that. It's a much less efficient system. It's like 2 ATPs instead of like 30 or something. So, very little, but they have to go through lots and lots of glucose that they take up. Then they produce a product that's extremely useful for the immune cells that are hanging out in the tumor environment. So the immune cells swarm into the tumor environment, but they don't fight the tumor. The tumor cells send them a signal, no, stand down. So they don't fight the tumor. And meanwhile, they're saying, oh, well, you're giving me all these fantastic nutrients. Why should I fight you? I'm going to just hang out here and enjoy the benefits of your fruits, So they've got, the glucose gets converted to, I'm trying to think now what it is.
Well, eventually lactate, right?
Lactate, that's the word. Yeah, I got overwhelmed with all the other words. Lactate, exactly. And that's something I knew from long ago. A famous thing that cancer cells do is make lots and lots of lactate and they ship it out. That lactate is a low deuterium nutrient because if you trace it back through the metabolism, it's a low deuterium nutrient. And it carries basically, it's a carrier of a proton that can get delivered to mitochondria as a low deuterium proton. And so the immune cells are happy to take up the lactate. And then they actually send it to the mitochondria and they use it to make ATP. And it's got that low deuterium proton that's so wonderful. And then, of course, the protons that are being shipped out into the medium are also low in deuterium because of that ATPase pump that won't. There was an article on yeast, their ATPase pumps. The article said that they very little or perhaps not at all did they send any deuterium out to that pump. That pump was so strong, they suspected it might not be able to send any deuterium out. That was really amazing to see that sentence in that paper. So the cancer cells are hoarding the deuterium. They're shutting down the mitochondria so the ATPase nanomotors don't get harmed by it because they're not working, they're taking a vacation. And meanwhile, they're getting their ATP from the glycolysis. They're sending out the lactate, which is another fantastic fuel. And then when you look at all kinds of other things that I've been rummaging through right now, I'm still learning. Almost everything I look at that's peculiar about cancer metabolism makes sense in the context of deuterium. It's truly amazing.
Wow, that is fascinating. And I want the audience to understand that I can't think of how any other cancer theory out there can explain why they would send their proton pumps to the surface of the cell to pump out protons. That is an active process. It's not like an accident. They are
Yeah, it seems
actively.
like it's a program that they've undertaken to to really fix a problem that's severe. And I'll tell you, when you treat cancer with all these poisons, that all these carcinogenic proteins, they give them chemicals that are carcinogenic to treat cancer, and often what happens is that cancer shrinks because it is being killed by these toxic chemicals. But meanwhile, it sends out some emissaries to metastasize to some other place, and now you're much worse off than you were before. So even though your main cancer shrank, you didn't solve the problem. The cancer is trying to desperately to send out colonies so they can take over on the project that the cancer is being foiled because it's got these toxic chemicals that are poisoning it.
But you're interpreting the cancer cells as providing a service to the body, right? So
exactly, they're gonna so their goal, and it's really fascinating how the cancer cells. And I was reading about this too before I knew about deuterium. How the cancer cells send out these signals that tell the immune cells to stand down. It's quite fascinating, and the immune cells then they don't attack the cancer. They're there, but they just sit around, don't attack. In fact, they actively support it. And the other cells that are in the tumor microenvironment, they're all there to support the cancer and help it to grow. And that's because the cancer is giving them back stuff they really, really need. And once those immune cells get enough good stuff from the cancer, they can become strong again. They're too weak to fight the cancer because their mitochondria are not working well because of all that deuterium. So the cancer is trying to rescue the immune cells. This is how I see it. To cure their problem with their mitochondria so that the immune cells are strong enough to then the cancer is happy to die,
Yeah, that makes sense. I like to take an Occam's razor kind of point of view with these theories, where I like to think that the simplest explanation is sort of the most likely. And so in my view, I just kind of took your theory to the logical conclusion, which is that the cancer cell is acting like a detox organ. And it's basically like a backup liver. I mean, the liver is the primary detox organ, but so if it is providing a service to the body, in other words, I guess what I'm trying to say is that it doesn't necessarily make a lot of sense for me for the cancer tumor to sometimes be good and sometimes be evil. It's either good or evil. And so I would say if we have a lot of evidence that it is being a good guy, why not just take that to the logical conclusion that it is good? And so
I totally agree. And I actually really like your theory, and I think it's true. I think it's not just deuterium. I think deuterium may be sort of the most prominent toxic exposure that the cells have. That's how I'm seeing it. But there are all these other things in our environment today, and the cancer is trying to hoard all the toxic stuff. It's sort of by turning off its mitochondria, it's more capable of dealing with, because a lot of these toxic chemicals affect the mitochondria, and that's the weak point. And you see mitochondrial dysfunction is associated with a huge list of diseases.
mm-hmm.
And the mitochondria become dysfunctional and there's all these chemicals that they've shown damage the mitochondria. A huge list, including glyphosate. I mean, that's well known. There's several papers talking about glyphosate damaging the mitochondria. They don't understand how it does it, but it does. Other chemicals, they understand how it does it. A lot of it's oxidative stress. Aluminum is really toxic. For example, lead, aluminum, mercury. I mean, mercury's whoa.
Yeah. No,
So,
actually, interestingly, I've found medical literature saying all of those toxic metals accumulate at high levels in cancer cells.
That totally makes sense. And I'm going to have to dig into that literature too.
Yeah. So like, for example,
to encompass yours.
like they've looked at, for example, breast cancer, and they found high levels of aluminum inside of breast cancer. And some people were thinking that it might be due to antiperspirants. So antiperspirants,
yes, I've read about that,
which are applied in the underarm, is very close to the breast tissue. So the idea would be that the breast tumors would form to sequester the aluminum from the antiperspirant.
And you could also, of course, say the aluminum got into the breast tissue and caused the cancer, right? They probably spin it that way as well, right?
Right. Yeah. And so what I like to push back against this sort of accidental theory by pointing to the fact that cancer cells upregulate their transport processes to basically concentrate toxins. And that's why the proton pump example that you gave is really compelling, because it's not passive, it's active. They are actively concentrating the toxins. And the same is true with some of these other toxins as well. They actively pull in the toxic metals.
Yeah, and it makes sense because what they've done, and it's true that even when because people are puzzled over that, even when their mitochondria are perfectly fine, they don't look like they're sick, but they don't use them to make ATP. They turn them off. So that's intentional, because that way they don't get harmed. They can survive a lot more toxicity because their mitochondria are turned off. Their mitochondrial ATPase nanomotors are turned off. That's a critical strategy that they take to protect themselves from toxicity. And then they can just load up on toxic chemicals and sequester them. And that keeps them away from the rest of the body. And that helps everybody else to be happy.
Yeah, that is such a brilliant insight. And it kind of pushes back against the metabolic theory of cancer. So Thomas Seyfried's metabolic theory of cancer. And I definitely deeply appreciate Seyfried's pioneering work. He really debunked the genetic theory, highlighted these nuclear transfer experiments, but he also really highlighted the key role of diet in cancer. But nevertheless, his theory of cancer is that mitochondrial damage leads to the Warburg effect, where they basically change their metabolism. And the mitochondrial damage is the key first step that leads to them behaving selfishly. But what you're saying and what I'm saying is that it's not a damage situation, it's a choice. It is a decision to not use the mitochondria when the mitochondria could be used, but it's actually mitochondrial oxidative phosphorylation is downregulated. They downregulate the process. And so it goes back to this idea that the body is brilliant, amazing, and strategic. Brilliant, amazing, and strategic. And it's not damaged, it's not faulty, it's not broken, which is the kind of standard mainstream view. They're always saying that we're broken and we're faulty. But anyway, what's fascinating is that I came at it from the angle, of course, after you. You really laid it out for deuterium beautifully, and I took your ideas and kind of ran with them. But I love how you reinterpreted the Warburg effect as like a way for the cell to act as a toxin storage vault. If it didn't change its metabolism, it couldn't act in that way.
Exactly, by virtue of safeguarding its mitochondria by turning them off, basically. Now they can afford to have these poisons that would wreck their mitochondria, but because those nanomotors are turned off, they're able to handle a lot more toxicity than a normal cell would.
Yeah,
and that's
absolutely.
Critical.
Yeah, that makes a ton of sense to me. I also looked at it from the perspective of glucose because, glucose does behave as a toxin when in excess. Of course, it plays a physiological
Yes, I saw that. I like that
right.
idea. Yes.
Yeah, so of course it plays a physiological role, but let's be honest, so many people nowadays are drinking sodas and eating candy all the time. And so many people are in glucose overload.
Yeah, and high glucose in the blood. Well, so cancer cells taking it out, so it's making it lower. It's working in the right direction.
Right, absolutely. Yeah. And so just as the cancer cells are trying to pull deuterium in, they're also pulling in the glucose. And of course, they do that intentionally. It's well documented that they upregulate their glucose transporters on their cell surface. They're pulling in the glucose. And then once again, this Warburg effect benefits not only from the deuterium side, but also the glucose side, because then they produce less reactive oxygen species.
And they produce lactate, which the neighbors love. The immune cells are very happy to take up that lactate. It's going to help them with their mitochondrial problem because it's a low deuterium nutrient.
Right, absolutely. So anyway, it's a beautiful solution to a multi-toxin problem, actually.
Yes.
And it helps with deuterium and it helps with glucose to do this Warburg effect of changing their metabolism.
And the aluminum, of course, it's taking up the aluminum.
Right. Yeah. So, and I'll just mention that, so did you have kind of a hunch like that this might extend to other toxins, like beyond? It sounds like you did, right?
Yeah. I even was thinking in terms of other toxins before I was thinking of deuterium, because I didn't know deuterium, before I knew deuterium. I've always suspected that cancer has I don't believe anything in the body happens without a good reason. I believe everything in metabolism happens for a reason. So I've always, in fact, I wrote something about Cancer to the Rescue before I knew about deuterium. I wrote an article about that in the Western Price Foundation's Wise Traditions Journal called Cancer to the Rescue. And I wrote that before I knew anything about deuterium. So I was already thinking in those terms. And I feel like, it was fascinating to me that the immune cells would swarm. They were drawn to the cancer tumor microenvironment. They're drawn to it. They go in there, but then they don't fight the cancer. So what's with that? I'm like, well, why don't they fight it? There they are. Their immune cells are supposed to, why don't they? They don't, because the cancer is giving them something they really need. and they're they're trying to recover, and the cancer is going to give them an opportunity to recover. And you know what they do with the chemical, the way they mess around with cancer with these various drugs that you take, the drugs can turn off they have drugs that can turn off the signals that the cancer cells make to shut down the immune cells, so that now the immune cells are, okay, I can fight the cancer now because they got blocked, right? And so now they fight the cancer. So they fight the cancer, and then the person ends up with a mess. They get a lot of autoimmune disease. They have to have another mechanism now to solve the deuterium problem because the cancer's let go. I mean, those immune cells are fighting the cancer when they don't really have the skill to do so because their mitochondria are sick. So then this just makes the immune system even sicker than it was before, because fighting the cancer is going to cause the immune cells a lot of suffering. They can't fight the cancer until they're strong.
Yeah, so that brings up, a deeper question about how the mainstream is thinking about cancer. And, I presume when you realize this new mechanism for cancer, did it change your worldview? Did it change how you think about mainstream medicine? And did it change how you think about how mainstream medicine treats cancer and what they're doing wrong? Yeah, how did that change?
It didn't change, because I have always, from long, long ago, even from my childhood, I would say, I've always been suspicious of pharma. And it's partly actually personal, very personal. I've never talked to anybody about this, but my mother was prescribed. But back in those 50s, there were a lot of doctors prescribing pharmaceutical drugs to treat depression, anxiety, those sorts of things. And the doctor prescribed uppers and downers, both. And they sort of said, well, when you need some energy, you need to vacuum the house, and take an upper. When you feel like relaxing, take a downer. Here's these uppers, here's these downers. And I don't even know what drugs they were, but they really messed her up. And it was a very sad thing for me to see. So I knew back then, I don't like these drugs, and I've always been, I've never liked statin drugs. I'm questioning blood pressure drugs right now even. So I really am starting to think that none of the drugs are good. I'm reaching a point where I'm completely, utterly against pharma.
Yeah, that makes a lot of sense. And definitely, when it comes to, it feels like cancer might be the worst situation because when I looked up, to see, there was this kind of unifying feature of toxins that they all create oxidative damage through reactive oxygen species.
Yes.
And then when I asked AI to make a table and rank the toxins based on how much ROS they produced, at the top of the list was chemotherapy.
Interesting. That's so interesting.
So
it never made sense to me to take a poison, for any reason. Why would you take a poison?
Mm-hmm.
And the statin drugs are poison also. I mean, I think most of the chemicals are poisons. And they're messing with biology because many of them are suppressing some transporter or some enzyme. And whenever you suppress an enzyme, you're messing up a system that the biology has perfected. And now it's got to go to plan B, and then plan C. I mean, it keeps on trying something else. And I think, you end up with dementia when you finally, when you treat heart disease, you end up with dementia. I mean, I think it's just, we have some epidemic in dementia right now. Mm-hmm. And I suspect that the pharmaceutical drugs are playing an important role there.
I definitely believe that. Yeah. So I mean, I already, like you said, didn't trust the mainstream medicine approach and big pharma. And then after looking into cancer, it's just become worse.
Exactly. I've become convinced that I'm totally right,
Yeah.
And I feel justified now. I feel very confident that I'm right. Whereas before I wasn't so sure,
Yeah.
Yeah. Now I'm very confident.
Yes. It looks like cancer may be the most mischaracterized disease out there in terms of how it's presented. I mean, other diseases are, like you said, dementia, Alzheimer's, that's a real tissue damaging disease where the toxins are just damaging the brain tissue.
Yeah, and you're protecting the brain by having cancer,
Yes.
Keeping those toxins away from the brain.
I even heard a clinician say once that she has clients that have brain tumors and other clients that have Alzheimer's, but she never gets clients that have both.
Yeah, I know. It protects you from everything else. That's what I've heard.
Right.
People will say, my mother's been living for decades with Alzheimer's and she's so healthy. I say, well, of course, because she sacrificed her brain.
Yeah.
Yeah.
Yeah. And So I'll just mention, like, we've talked about a couple of different cancer theories. We did talk about, well, the mainstream theory, which the nuclear transfer experiments kind of, debunked. And then we talked about the metabolic theory of cancer, which we've been trying to push back on because it looks like the toxins aren't getting in by accident. They're actually pulled in. There's a couple other random theories that I see in alternative medicine. There's the parasite theory of cancer.
Yeah.
There's a fungal theory of cancer. Some people are talking about emotional trauma. And I just want to mention that we had, like, there's mainstream medicine, which I've kind of given up on, but there's a space that I feel like we have an opportunity for, which is alternative medicine.
Yes.
And if we can make progress in getting our theory into alternative medicine, then maybe we can save some lives. But we are fighting in that space things like the parasite theory, the fungal theory, and emotional
Yeah,
trauma.
now I will say, I suspect that parasites and fungus also serve a similar role. And so if you've got a lot of toxicity, you're going to get a fungus problem. And then maybe the fungus problem isn't good enough to solve the problem. So now you end up with cancer as well because you've got so much toxicity. It's just an overburden of toxicity. The fungus is an indicator that you've got toxic exposures. And so is the cancer, but these exposures are so bad, the fungus can't do it by itself. So it's sort of, the cancer comes along as well to help out with the problem because we have so much toxicity in the modern world.
That is a fascinating point, and I want the audience to really listen to what Stephanie just said. You are basically saying that these microorganisms can also play a similar role to the cancer cells in being protective against toxicity. Can you just elaborate a little bit further on that? Yeah.
Well, in fact, those yeast, I mentioned those yeast ATPase pumps that are in yeast. And the yeast can pump, that's the article about the yeast that said they couldn't pump out deuterium, they could only pump out protons. So when they pump out protons with those pumps, they end up enriched in deuterium internally because they've let go of protons. And yeast can actually handle quite a bit of deuterium. And actually, there's a fascinating article about bacteria, and I forget which strain, I think it might have even been E. coli. I think it was. There were these tanks that were storing deuterium. It was deuterated. It was something from the military. I think it was very rich in deuterium in a tank. I forget exactly what it was. I need to go back and look. But they found, and it was sealed off, so there was no oxygen. And they found there were E. coli living in there, and they had incredibly high deuterium. Their water was basically D2O. I mean, it was like pure deuterium water. And they were able to live on that. And they showed then that you could actually gradually enrich the water with deuterium in an E. coli culture, and they could adjust over time. If you hit them suddenly, they would kill off, but they could gradually acquire a skill of tolerating extremely high deuterium levels in their water supply. So it's clear that the microbes are able to sequester deuterium. And when the microbes are making deuterium-depleted nutrients, well, they're going to have more deuterium internally when they do that. Anytime you're making something that's low in deuterium and giving it to the world, you're going to be high in deuterium. So I think all the microbes are probably have more deuterium in their water than the body does. They're all sort of sequestering deuterium to some degree, and at the same time producing these deuterium-depleted nutrients. And I want to say, the cancer that I wrote a paper recently where we showed cancer cells reacting to. There were studies that showed if you give them deuterium-rich water, you grow the cells in culture with water that has twice as much deuterium, so it's 300 instead of 155 parts per million, and you expose them to that water, they end up upregulating almost a huge number of, I think, like 200 different enzymes that are associated with cancer. So they upregulated all kinds of enzymes that in humans are associated with cancer. And when you gave them deuterium-depleted water, it caused them to downregulate two enzymes that are associated with that cause increased risk to cancer. In other words, it was the exact opposite. It was only two, but they were critical ones. It was Mike, and I forget what they were. I don't know, but there were these two. C-Mike was one of them, I think. Two molecules that are well known to be cause a cell to become a cancer cell. tumorigenic, I should say, tumorigenic, downregulate, and also cause oxidative stress and cause the cells to die. So this was the deuterium depleted water. So they, both the deuterium depleted and the deuterium excess water applied to cancer grown in culture are consistent with the idea that deuterium, that cancer cells hoard deuterium.
Mm-hmm. Yeah, no, this is fascinating. So just to recap for the audience there, so some people have connected things like funguses and parasites to cancer, but once again, it looks like correlation, not causation, and that basically it's the underlying root cause appears to be toxicity, toxic overload of the body. And then these other species are there to kind of clean up the mess and hoard these things. They play a very similar role to the cancer cells themselves. So that is absolutely fascinating. Yeah, so thank you so much today for talking to me. I feel like we've covered a lot of ground. And so I just want to say, once again, for the audience's benefit, that Stephanie's work was the key inspiration for my own theory of cancer, which is called Toxin Sequestration Theory, or TST. And all I've done is I've taken the mechanisms that Stephanie has laid out for deuterium and just extend them to other toxins. So I wanted to, deeply express my gratitude to Stephanie for her pioneering work. I do believe she should be getting a Nobel Prize for this. It really is completely changing the script, flipping the script on what cancer is doing. And I'm hoping that, well, actually one more question would just be, is there anything else that we can do you think, that would kind of change people's view on tumors? How else can we change the public's view on tumors? Is there anything else we can do or is it just too...
I can tell you how I think that to live a healthy life, an important aspect is getting out in the sunshine and getting grounded. So the electricity is important in the body, and walking on the ground where you're getting negative charge from the earth is important, and getting the sunlight. Sunlight is incredibly healing. Don't use the sunscreen. Don't wear the sunglasses. I mean, this is my belief. Sunlight is very, very healthy, and being outdoors, and even being outdoors in the woods, even you're breathing what the trees are releasing, that's probably beneficial. I mean, it's just like in nature. Hang out in nature as much as you can, and outdoors, and walk in the shallow water at the shore of the ocean. If you happen to live near the ocean, extremely healthy.
Yeah, absolutely. Great advice. People can, of course, find more about Stephanie on social media. You're on Twitter, you're on Substack. I subscribe to your Substack, and we can provide links to your social media profiles.
That'd be great.
In the description. So thanks for this wonderful conversation. I learned a lot, and once again, thank you for the inspiring research that you've done.
Thank you so much for having me. I really enjoyed it.
All right, I'm going to end the recording. Thanks.